Gene regulation. Gene regulation is the concept that the expression of many genes are regulated, meaning that they're not expressed at the same levels at all times. Instead, there are mechanisms by which the expression levels of the genes change. The majority of genes within a cell are regulated. There are many different cellular processes which are regulated, some of which are shown here. But there are also genes within a cell that are not regulated, and we often will refer to those genes as constitutively expressed, meaning that they're expressed at the same levels regardless of circumstances. The mechanisms by which regulation of genes occurs, there are a couple of different mechanisms, which will be shown on the next slide. So what's indicated here is that the expression of a gene is defined as a gene is transcribed, and most of the time it is translated into a protein. Sometimes things happen to that protein after it is translated, so this is a post-translational modifications that will eventually lead to a functional protein. And at any step along the way, you can alter the potential activity or function of the gene product. So, for example, in transcription, you can regulate transcription through control of whether or not transcription is initiated. There's another process we will talk about in bacteria in particular that's called attenuation, and that involves whether or not transcription is terminated early or whether it is allowed to proceed during the elongation phase of transcription. You can also have gene expression regulated at the translational step, whether or not an mRNA is translated. And then you can also have post-translational modifications that when, for example, if you can covalently modify a protein, or you can non-covalently have other proteins bind to it that have the ability to impact its function as well. But for the purposes of this slideshow, we're really going to focus in on this step here, which is the regulation of transcription. So, transcriptional regulation, and just like we saw for replication, transcription, translation, we're going to focus in on bacterial examples. And in bacteria, the most common way that genes are regulated is at the transcriptional level. Regulation of RNA synthesis or transcription can be either a positive or a negative form of regulation. So that means that the rate of RNA synthesis can either be increased or decreased. The primary mechanism by which this is achieved is through the interaction of regulatory proteins. So you have regulatory proteins which will bind to the DNA in and around the promoter region, which then have an impact on the ability for RNA polymerase to initiate transcription. A class of proteins that binds to DNA and decreases levels of transcription or inhibits transcription are referred to as repressors. And then a class of proteins which increase transcription levels are referred to as activators. So for once we have things that are named in a very straightforward way. Collectively, the proteins which will bind to DNA and influence transcription levels, we refer to them as transcription factors. And again, they can either be positive transcription factors or negative transcription factors, meaning that they can either increase or decrease transcription levels. And these transcription factors themselves can be modulated, their activity can be modulated by their interactions with small effector molecules. And these small effector, what I mean by a small effector molecule is they're typically organic compounds which have the ability to non-covalently bind to the transcription factor. They induce an allosteric change in shape, and that change in shape is going to interconvert the protein between an inactive and an active state, and only in the active state can that protein bind to DNA efficiently and modulate transcriptional activity. So these small effector molecules do not bind to DNA directly. Instead, they are binding to the regulatory proteins, which then, depending on their state, can bind to DNA. So negative control of the transcription of RNA involves the presence of repressor proteins. The repressor proteins will bind to a regulatory site. When it is the regulatory sites that bind repressor proteins are often also referred to as operators. And the repressor protein, as we mentioned before, has at least two different conformations, an active conformation and an inactive conformation. And the transformation between these two forms is mediated by the presence of the small molecule effectors. And in this case, we have very specific names for the small molecule effectors. We have inducers, right, which are small molecule effectors when the presence of the small molecule effector increases mRNA synthesis, and co-repressors, which have the opposite effect. The presence of a co-repressor will decrease mRNA synthesis levels. So let's look at an example here. We have a strand of DNA. The yellow blob represents the transcription factor. In this case, it's a repressor protein. The repressor protein recognizes the operator, that's the control site, which is in the proximity of the promoter. That's the site that's recognized by RNA polymerase for the initiation of transcription. So when the repressor protein binds to DNA, it prevents transcription from occurring. Now, this repressor protein will only bind to the DNA in its apo form, which means the absence of any cofactors or any small molecules that might bind to it. So when a cofactor is present, or this small molecule effector molecule is present, you have a change in conformation of the repressor protein. It no longer binds to the control site, the operator, freeing up the promoter to be accessed by RNA polymerase, and you get transcription of this gene, right, which is downstream of the two control sites. Because the small molecule is increasing transcription levels, we refer to it as an inducer. Alternatively, we have a scenario where everything looks the same, but now the apo form of the repressor does not have the ability to bind to the DNA. So in the absence of the small molecule effector, we have efficient transcription that occurs. But in the presence of the small molecule effector, we now have a change in conformation which efficiently binds to that operator region. So the entire repressor protein can now recognize DNA, efficiently bind to it, preventing efficient initiation of transcription at the promoter site. In scenarios where genes are positively regulated, so that means that there is the activity of an activator protein, we have two kind of analogous scenarios. So the graphical scheme is the same. This represents the transcription factor. In these cases, it will be an activator protein. Here is our DNA. The green region is still the promoter, and the control site is referred to as an activator binding site rather than an operator. Right. So we have an activator binding site, and we have a promoter that are part of the DNA, and our gene resides downstream of them. In the absence of a small molecule effector, in this scenario, the activator protein is in its inactive conformation, so it does not efficiently bind to the binding site, and therefore you have relatively low levels of transcription because the activator protein will increase levels of transcription when it interacts with DNA. In the presence of the small molecule effector, shown as this blue kind of diamond, the activator protein assumes a different conformation and now efficiently binds to that control site, the activator binding site, allowing higher levels of transcription to occur. So once again, since this presence of this small molecule increases or induces transcription, we refer to it as an inducer, even though the mechanism is very different than what we saw for the previous inducer. Alternatively, you can have the apo form of the activator protein is the one that binds DNA and allows to very high, relatively high transcription levels, as is shown in this scenario. The presence of the small molecule effector creates an inactive form of the activator protein, leading to lower levels of transcription. And in this case, that presence of that small molecule effector decreases transcription levels as a result, so we refer to it as an inhibitor. I've included this slide basically as a summary in text of the main points of the narration from the previous slides that you can review at your convenience. If we look at a map of a typical gene, as is shown here, here's a piece of DNA. Right? There are several key features on the DNA. The promoter, which is where transcription is initiated. We have a terminator, which is where transcription is ended or terminated. And now we have these control sites as well that can either be binding sites for repressor proteins or activator proteins that are going to regulate the levels of transcription of these genes. And then you have, of course, the coding region, which includes a translational start site, a translational stop site, so a start codon and a stop codon, which are where translation is initiated and terminated, respectively, that allows the coding sequence to be transcribed into messenger RNA and then translated into a protein. The two examples we'll be looking at in more detail to understand the nuts and bolts of gene regulation are actually operons. So to define an operon is a regulatory unit that has more than one gene that's under the control of a single promoter. Operons are mainly found in prokaryotes, so bacteria and archaea. There are some examples of operons that are found in eukaryotic systems, but the fundamental way that they operate is a bit different than what we'll see for the prokaryotes. So the examples that we'll give here and the definitions that we'll give are essentially for prokaryotic operons. Eukaryotic operons are sort of rare and a little bit of a departure from the norm as opposed to prokaryotic operons, which are very common. An operon will typically lead to the production of a polycistronic mRNA, which is defined as an mRNA which has multiple open reading frames or coding sequences on a single strand of messenger RNA. And in bacteria, it's very common that the individual proteins that are encoded by the genes on this polycistronic mRNA are functionally related to each other. So the two examples we will look at, they all encode enzymes, which are part of a biochemical pathway. So they're individual steps in the biochemical pathway. Because they're coordinated like this, the bacterium can then regulate an entire group of genes that are part of a single process with a single regulatory apparatus. There's a vocabulary term here, structural gene, and that refers to a gene which leads to a protein. It can really be almost any kind of protein except for a regulatory protein. So a gene that leads to a regulatory protein is not considered a structural gene. Almost any other protein encoding gene is a structural gene. But in the two scenarios we're going to look at, the genes all encode enzymes. So the first major example we're going to look at comes from our friend E. coli, and it is referred to as the lac operon. And the lac operon, lac is short for lactose. Lactose is a sugar. And from the perspective of the lac operon, lactose is a fuel source. So The lac operon is going to encode several catabolic genes, which are involved in the breaking down of lactose into simpler molecular units that eventually be used as an energy source for the bacterium. So shown here is a map of the lac operon as well as a second regulatory gene. So the lac operon extends from here to here. It has many of the elements we've come to expect. It has a promoter. It has a terminator. It has 3 structural genes, LAC-Z, LAC-Y, and LAC-A. And it has 2 different controlling sites. It has an operator, which is where a repressor will bind, and it has a cap site, and cap stands for the catabolic activator protein. So this is an activator binding site. There is a second gene here that's shown, which is a regulatory gene, and it is, we'll get to that in a later slide. But the 3 different structural genes, LAC-Z, LAC-Y, LAC-A, as I previously mentioned, all encode enzymes, beta-galactosidase, lac-permease, and transacetylase. And it should be pointed out that this transacetylase we'll kind of ignore for the purposes of this slide because it is an enzyme which the function of it is known. It covalently modifies lactose, but the functional necessity of this enzyme isn't really clear. And when you kind of delete this enzyme experimentally, that doesn't seem to have a major impact on the functioning of the organism or the operon. So, the lac promoter is an example of a so-called or quote-unquote leaky promoter. So think about what this term might imply and see if you can come up with an answer before you go to the next slide. So, a leaky promoter, what is implied by that term is a promoter which allows low levels of transcription to occur even when there isn't any induction signal. So the best answer for this is B. And a conceptual analogy is think of it as a leaky faucet, right? You have the ability to change the levels of water that come out of a faucet by turning a knob. And it's kind of a useful way to think about gene regulation. You have these kind of volume knobs or flow knobs that are under the control of these molecular mechanisms. But when it's a leaky promoter, even when you turn the knob all the way down or you turn the faucet handle all the way down, you still get low levels of expression. So we're going to look at a little bit more detail of the functions of the 3 structural genes of the lac operon. The genes are LAC-Z, LAC-Y, and LAC-A. They encode beta-galactosidase, lac-permease, as well as this transacetylase, but we're basically going to ignore the transacetylase from here on out. So this picture represents a bacterial cell. It's kind of a cartoon of a bacterial cell. The lac-permease is shown here. It is a membrane channel, so it is what allows the importation of lactose. So if lactose is outside of the bacterial cell, which is shown here, it's exogenous, it allows that lactose to be imported to the interior of the bacterium. And remember, this is a prokaryote without any individual compartments. There are no organelles within a bacterium. And so once it's in the cytoplasm, it can be accessed by any of the enzymes in the cytoplasm. And in particular, the enzyme that is going to interact most with lactose is beta-galactosidase. So what's shown here is a chemical representation of lactose. You can see it is a disaccharide. 2 different simple sugars are linked together. Beta-galactosidase actually catalyzes a couple of different reactions, which sometimes happens with enzymes, even though enzymes are very specific. This enzyme uses the same or a couple of different substrates and catalyzes a few different reactions. So the main reaction that beta-galactosidase catalyzes is the cleavage of these 2 individual monosaccharides from the disaccharide. So the main reaction is that you end up with 2 simple sugars, galactose and glucose, as the breakdown products of lactose. But a small amount of a side reaction can also occur, whereby which essentially you break this bond and then it reforms, and you end up changing the stereochemistry of the linkage between the two individual monosaccharides. So you end up with an isomer of lactose that's referred to as allolactose. And this is an important molecule because allolactose is going to be the small molecule effector that interacts with the repressor protein that regulates the lac operon. And allolactose can further be broken down by beta-galactosidase once again into the two simple sugars. So a bit more detail about the regulatory gene that's shown on this map. It's also known as the LAC-I gene. Technically, it is not part of the lac operon proper. It is a separate gene, has its own promoter. It would have its own repressor that's shown here. Usually there's a little bit of a gap between these two. For some reason this map isn't showing it, probably just for space constraints. The regulatory gene, the promoter for the regulatory gene is what's known as a constitutive promoter. And what that means is that that gene is expressed at constant levels. So regardless of circumstances within the cell, changing conditions, this gene will always be expressed at the same level. The product of the gene is known as the lac repressor protein. And just as a reminder, proteins within cells are relatively unstable, fragile entities, meaning that they degrade over time. And so, as a result, in order to have a continued presence of that functional protein within a cell, you have to continue to manufacture it, continue to express it. So this constitutive expression of the LAC-I gene product means that you'll have constant levels of a lac repressor within cells. Although they will be at fairly low levels. This is a relatively weak promoter, which means that it's not expressed at an extremely high rate, it's just expressed at a constant rate, meaning that there's probably not that many molecules of the lac repressor protein, maybe about 40 in the cell at any given time. Now, the lac repressor actually functions as a tetramer, meaning that 4 identical subunits of the gene product combine non-covalently to form a functional lac repressor complex. And this is based on structural studies what the lac repressor complex looks like. Here's the DNA. It's being looped around. We have binding sites at 2 different locations where the lac repressor interacts with the DNA. The promoter is actually contained within the loop here. So you can see why when the lac repressor complex is bound to DNA, RNA polymerase would have a hard time accessing the promoter. So, as we mentioned, the lac operon is regulated 2 different ways. Both are regulated at the transcription initiation level. The first methodology involves that repressor protein from the earlier slide. That's a negative form of repression. There's a second form, which we'll talk about a little bit later, that is positive regulation, and that means that it is regulated by the presence of an activator protein. So we'll spend a bit of time talking about each of these 2 different methods of regulation, starting with the first one, this negative regulation. And as we've kind of mentioned earlier, this is inducible and a negative control mechanism, meaning that because it's negative control, a repressor protein is involved. That's the lac repressor protein we saw previously. The inducer is allolactose, which we've also mentioned previously. And the interaction of these are such that the lac repressor protein will only bind to DNA in the absence of lactose or allolactose. And in the presence of allolactose, the lac repressor undergoes a conformational change, which inactivates it, meaning it cannot bind DNA and therefore cannot repress expression of the lac operon. Here is the mechanism, that negative control mechanism, in kind of a pictorial format. This line represents the DNA. Here are the 3 lac structural genes, Z, Y, and A. This is the operator region, the region that is recognized by the repressor protein. Here's the promoter, which is bound, which is where RNA polymerase recognizes and initiates transcription. And over here is the LAC-I gene, which remember is constitutively expressed. So it always produces a steady amount of the LAC repressor protein. And in the absence of lactose, this LAC repressor protein will bind to DNA and therefore downregulate expression of the LAC operon genes. Alternatively, when lactose is present, the LAC-I gene is still constitutively expressed, but the presence of lactose means that there is some allolactose present through the activity of, as it gets converted by the activity of beta-galactosidase, which remember is always expressed at a low level, right? It has that leaky expression from the LAC promoter. It's not really shown here, but this is really allolactose that binds to the LAC repressor, inducing a conformational change which allows much higher levels of transcription of the LAC structural genes. And they can be transcribed and therefore translated as ribosomes access the polycistronic mRNA. So these little blobs represent ribosomes, and then these represent the individual polypeptides that are the functional products. All right, so this next slide is just another representation of the same thing from a different textbook that kind of shows it using slightly different graphics. So we start in an environment where there is no lactose, therefore there is no allolactose. This is our piece of DNA, the three structural genes again, the operator, the promoter, and the LAC-I gene. And as before, the LAC-I gene is constitutively transcribed and translated into the LAC repressor protein. And you can see here, there's a little bit more structural detail of what's going on. This representation of the LAC repressor protein is a little closer to what we know that it looks like based on structural studies that have been done. So this is kind of, you can see there's 4 individual kind of mirror image subunits that are shown here, and it looks kind of like 2 cartoon characters shaking hands or kissing or something. But in the absence of lactose, and therefore allolactose, this LAC repressor is active, and it can bind to the operator site. And when it does this, it denies the ability for the RNA polymerase to access the promoter, and as a result, you get much lower levels of transcription of the various LAC structural genes. So this is the repressed state. When lactose is present, we now have allolactose. Our LAC-I gene still is constitutively expressed, but because allolactose is present, it is now in its inactive form, no longer binds to the LAC operator, which now means that the LAC promoter is accessible to the RNA polymerase, and we have an induced state where we have transcription of the polycistronic mRNA and result in translation of the various LAC structural genes, right? Beta-galactosidase. Yet another representation of the same process. Here's our DNA, LAC structural genes, operator, promoter. Here's our LAC-I gene. Again, it has its own promoter and its own terminator. It is constitutively expressed. In the absence of lactose, we're now showing it as kind of like this arch-shaped building block. It will bind to the operator, and this is the repressed state. In the induced state, we have allolactose present, conformational change in the LAC repressor, but again, it's still expressed constitutively. And as a result, we get higher levels of expression. The nice thing about this representation is it shows the coupling of transcription and translation. So you can see that this polycistronic mRNA hasn't completed being transcribed, right? Transcription hasn't terminated yet. It's still being spooled out of the RNA polymerase. But even then, it's nascent or still forming polycistronic mRNA is actively being translated by multiple ribosomes. Okay, so multiple ribosomes on a single piece of RNA is a structure that is referred to as a polysome. And we have multiple ribosomes all translating the same open reading frame and generating lots and lots of gene products. All right, one final representation of this lac operon for those of you that prefer looking at things left to right rather than top to bottom. Here is our original piece of DNA, our 3 lac structural genes, operator, promoter, and our lac I gene is shown here with its own promoter. Right. So if we look at these two scenarios side by side, absence of lactose versus presence of lactose, when no lactose is present, the constitutively expressed lac repressor binds to the operator, inhibiting transcription. When lactose is present in the cell, because there is some basal level of expression of the lac permease, which is the product of the lac Y gene, that lactose gets inside the cell. The basal levels of expression of beta-galactosidase will convert most of that lactose to glucose and galactose monomers, but some of it will be converted to allolactose. And even small amounts of allolactose will bind to the lac repressor proteins, because there aren't very many lac repressor proteins in the cell. That will prevent the lac repressor protein from binding to DNA, and therefore we have induction, and the induction will allow transcription to occur at a much higher rate, meaning lots more gene product and lots more import of lactose over time. So for the next slide, I find that it's helpful if you kind of anthropomorphize the bacterial cell. So imagine that you are the bacterial cell, right? And we're going to go through the cycle of the lac operon induction and repression. So imagine you are a bacterial cell in an environment where there is no lactose, right? So imagine you're just kind of hanging out inside a puddle on the sidewalk of the Excelsior Concourse. This is the bacterial cell. This is your DNA. It's circular in bacteria, whether it be a plasmid or the bacterial chromosome. The lac operon resides on the DNA, and it is expressed at low levels because it has a leaky promoter. So as a result, you'll have small levels of lac permease, small levels of beta-galactosidase, and small levels of that transacetylase, all of which will be present in the cell, although at kind of relatively low levels. Now, imagine your environment suddenly changes. Right? Somehow, your puddle becomes inundated with a large supply of lactose. I like to think of it as somebody has dropped an ice cream cone in your puddle, right? A food that's rich in lactose. Now, because there is some levels of lac Y permease out in your cell membrane, you're going to start importing lactose inside the cell. Because there are some basal levels of beta-galactosidase inside the cell, you are going to convert most of that to galactose and glucose, but some of it will get converted to allolactose. And that allolactose will interact with the lac repressor protein, and when it forms a complex, it will release from the DNA and allow expression levels of the lac operon structural genes to increase, which we see here. So now, because expression levels have increased, we get a lot more lac permease inside this, embedded within the cell membrane. We have a lot more beta-galactosidase, so much more of the lactose gets imported into the cell, gets converted into lactose and, it gets converted into glucose and galactose, and it, a small amount of it gets converted into allolactose, so you kind of have this feed-forward cycle where the more lactose you import, the more gene products you generate. Eventually, all of this lactose will get imported and consumed by the bacterial cell. And any remaining allolactose can also be broken down and converted into glucose and galactose and all consumed as part of food. So the exterior levels of lactose, the exogenous levels of lactose will drop. And now you'll have a cell where you have lots of these gene products, but no more lactose outside the cell that has the ability to be imported and utilized. Now, since these proteins are unstable, they will degrade over time. There's no allolactose present in the cell, so the repressor protein, which is constantly being replenished by its constitutive promoter, will rebind to the DNA, And as a result, you will return to that initial state. So you can think of this as kind of a cyclical response. There is a second mechanism of regulation, which we mentioned previously, that is a form of positive regulation. And this is mediated by an activator protein. And collectively, the purpose of this mechanism is to allow bacteria to make food choices. So the logic is as follows. And you have an environment that has multiple types of sugars, right? So imagine we have both glucose present and lactose present. E. coli will preferentially utilize glucose first because it requires fewer gene products in order to be catabolized. So you can think of it as kind of a food preference. This might be, for those of you that are carnivores, this might be bacon and this might be tofu, right? And you're in an environment that has both bacon and tofu, you may prefer bacon over tofu. And same kind of concept is going on here with the bacteria. So this this positive regulation that is mediated by an activator protein is also known kind of confusingly as catabolite repression, which is the idea that a catabolite is going to effectively repress pathways that are going to allow this choice between glucose over lactose when both are present. So the molecular details of how this works, there is an activator protein that's called the catabolite activator protein. It's called the CAP protein also. And it has other names as well, the cyclic AMP receptor protein you may see in some texts. I'll refer to it as CAP in the slides that follow. The small molecule effector of the CAP protein is a molecule that's known as cyclic AMP. Cyclic AMP is derived from ATP. It involves an enzyme that's called adenylyl cyclase. So this adenylyl cyclase is able to convert ATP into cyclic AMP. So they are both nucleotide monomers. ATP, as we know, is a major source of energy within the cell. Cyclic AMP is a signaling molecule. And so not all ATP is converted into cyclic AMP, only a very small amount of it. But cyclic AMP concentrations are going to be relative to ATP concentrations. So as a result, cyclic AMP concentrations are going to give a readout as to what are the energy levels within a cell. So the logic is as follows. Glucose is a negative regulator of adenylyl cyclase activity, meaning that when you have high levels of glucose, it inhibits the activity of adenylyl cyclase, which is represented as this arrow. So this transformation is a chemical reaction. ATP is converted into cyclic AMP, and most of these chemical reactions that occur within cells require enzymes to accelerate those reactions, and the enzyme that accelerates this reaction is adenylyl cyclase. Glucose inhibits adenylyl cyclase. So when you have high levels of glucose, you get no adenylyl cyclase activity, and therefore cyclic AMP levels within the cell are low. When you have relatively low levels of glucose, adenylyl cyclase is not inhibited, meaning that you can easily convert ATP into cyclic AMP. So there is an inverse relationship between the concentration of glucose and the concentration of cyclic AMP. When cyclic AMP is present, it can form a complex with the CAP protein, and this complex is now the active form of this activator protein, so it can efficiently bind to the activator binding site. And when it does that, it will actively recruit RNA polymerase to the promoter site, thereby increasing transcription levels of the lac operon. So, another picture of it, the CAP protein actually functions as a homodimer, so two identical subunits of the CAP protein interact with each other non-covalently. When cyclic AMP is present, a single cyclic AMP molecule binds at the interface between these two monomers, forming an active complex. This active complex binds to the cap binding site. That allows the RNA polymerase, shown here. Here's the sigma factor interacting with the promoter region. So it recruits RNA polymerase, and then RNA polymerase has the ability to actively transcribe the lac operon genes at a much higher rate. So let's stop for a minute and consider a conceptual question. So what we're asking is, what are conditions of the choices below where we would see the highest level of expression of the lac operon? So we know that lactose can be converted into allolactose and acts as an inducer. So the highest levels will be conditions in which lactose is present. We know that glucose is inversely related to the concentration of cyclic AMP. So that, and cyclic AMP also acts as an inducer. So when both lactose is present and glucose is absent, therefore there's a high concentration of cyclic AMP, we'd expect to see the highest levels of transcription within the lac operon. So let's look at these two control mechanisms working in conjunction in a little bit more detail. So the first scenario is we have glucose is present and no lactose. So in this scenario, the lac repressor protein will bind to the operator, right? And the cap protein will not bind to the activator binding site. So this is the scenario where we would expect to see the lowest levels of expression, right? We are being repressed and we are not being activated. There will still be some basal level of expression because it is a leaky promoter, but we would expect the lowest levels of expression to be in scenario A. In scenario B, we have glucose present, and lactose present. Meaning, so both sugars are present, right? That means that we have no repression occurring, right? Because allolactose is present, and therefore we have no repression, but we also have no activation. So in this scenario, we would expect some sort of intermediate level of expression. We would expect it to be higher than scenario A, but it's going to be lower than maximal expression. So maximal expression is going to occur in scenario C, where we have no repression, but we do have activation. No glucose is present, meaning cyclic AMP is present. We have activation, no repression, and as a result, we see the largest level of expression of the lac operon. So another representation, graphical representation of the way that these two control mechanisms work with respect to each other. In scenario A, we have presence of cyclic AMP and lactose, which means that the repressor will not repress because it is inactive. The activator will activate because it is active. And as a result, this is the scenario that was seen in the previous slide where we have maximal levels of expression. When we have lactose present and no cyclic AMP, this means we have no repression, but also no inactivation. Both of the transcription factors are inactive, so nothing is interacting with the DNA. As a result, this is that intermediate level of expression. Third scenario, we have cyclic AMP and no lactose. This means that we are getting both repression and activation at the same time. Both transcription factors are interacting with the DNA and basically fighting with each other. They have opposite effects. This is going to repress, this is going to activate. So as a result, we'll see kind of another intermediate state. This scenario was not shown in that previous set of graphics. And then the fourth scenario, where we have no cyclic AMP, no lactose. As a result, we get repression, no activation. This is the lowest possible level of transcription and expression of the lac genes. The second example that we're going to look at, a completely different operon, also from our buddy E. coli, is known as the trip operon. And trip is short for the amino acid tryptophan. It's the three-letter code that is equivalent to tryptophan. Tryptophan as an amino acid is important for the construction of nearly all proteins, so it's an absolutely required building block that needs to be present in any living cell in order to make proteins. And so the logic of the trip operon from a metabolic point of view is going to essentially be the opposite of the lac operon. Remember, lactose was a potential fuel source that needs to be broken down, so the object was to catabolize lactose, to basically have the cell eat the lactose. And the trip operon, the objective is to make tryptophan, using an anabolic pathway. So using simple precursor molecules to build a more complex amino acid tryptophan. Now, you've probably heard of tryptophan before in terms of turkey and getting sleepy. In the context of human health, tryptophan is what's called an essential amino acid, meaning that we and most animals have to get tryptophan through our diet. But bacteria and plants have the biochemical pathways that allow tryptophan to be made. So even though we cannot make tryptophan, bacteria have the ability to biosynthesize their own tryptophan. This is a map of what the trip operon looks like. There are 1, 2, 3, 4, 5 different structural genes, trip A through E. They all encode enzymes that are involved in the biosynthesis of tryptophan from simple precursors or simpler precursors. The operon has its own promoter, its own operator. It has this funny leader region that includes an attenuator, which we'll talk about a little bit later in more detail. But the trip operon is regulated using 2 different mechanisms. The first mechanism is going to be very similar to what we saw for the lac operon, and it's going to involve the trip repressor protein, which is a separate gene that has its own separate constitutive promoter. And then there'll also be this trip L, this leader region, which we'll get into a bit later, that is involved in a mechanism of regulation that is known as attenuation. If we look at the trip operon in a bit more detail, the trip mRNA is going to be a polycistronic mRNA. It's going to include the coding regions of all 5 of the structural genes, as well as this leader region, which we're going to ignore for the time being. We'll come back to that. Each of these encodes an enzyme. These enzymes are involved in catalyzing various reactions that allow relatively simple precursors, corsmate, glutamine, PRPP, and serine, to be combined to form a bit more complicated molecule, tryptophan. So tryptophan is the largest of the amino acids. It's also the most, even though it's used in essentially all proteins, tryptophan is the rarest of the amino acids, meaning it's the least common within cells and it's the least utilized within proteins. So the logic of this first form of regulation is going to involve a repressor protein, and this repressor protein is going to be modulated by a small effector, and that small effector is going to be that amino acid, the product of this pathway, which is tryptophan. So if you think about the logic of this, the purpose of this operon is to create tryptophan. So it really only wants to function when tryptophan levels are insufficient. Otherwise, you're wasting energy creating tryptophan if you have abundant tryptophan already present within the cell. So it's going to sense low levels of tryptophan and allow expression of the trip operon genes. If there's high levels of tryptophan, it's going to repress expression of these genes. So here's a map of what is happening. This represents the piece of DNA. Here are the trip structural genes. This gene, the trip L, is actually a regulatory protein, so it's not considered a structural gene. Again, we'll get into that in a little bit later. The trip repressor protein is a separate gene found elsewhere within the genome of the bacteria. It is constitutively expressed to create an inactive trip repressor. Because tryptophan levels are low, right, there is no small effector that activates this trip repressor protein, and that allows efficient expression of the trip operon. And all of the various trip structural genes are transcribed. When tryptophan levels are high, the trip repressor protein is still constitutively expressed, but because tryptophan is present, it will bind to the trip repressor protein, thereby activating it, allowing it to bind to the DNA, and repress transcription of the trip operon. Right. So this is that first negative control mechanism. There is a second control mechanism. So that first control mechanism is not an absolute control mechanism. It doesn't completely shut down initiation of transcription of the trip operon in a similar way that we saw for the lac operon. So there is a second control mechanism known as attenuation that will operate when tryptophan levels are high, that will operate at the level of transcriptional elongation. So attenuation means you have a premature termination of transcription. So the transcription does not proceed all the way through the structural genes. Instead, it is terminated early on after initiation. The way that the attenuator works is that when you have high levels of transcription, if some initiation of transcription occurs, right, so the RNA polymerase is able to initiate transcription, it gets past the trip repressor protein, or the trip repressor protein doesn't have 100% residency on this space, so maybe a small percentage of the time that it isn't present, you get initiation of transcription. This attenuator sequence is going to facilitate the premature termination of transcription. And this is that second mechanism of regulation. So just like the lac operon, we have two alternate mechanisms that have the ability to downregulate or control the levels of expression of the trip operon structural genes. So this is an idea of how attenuation occurs, and it all involves sequences within that leader sequence. So you have a molecule, a single-stranded piece of RNA that represents the mRNA transcript of the lac operon. You have a very short gene that encodes a handful of amino acids. Right? It has its own start codon, it has its own stop codon. This gene is referred to as the TRPL gene, right? Also known as the leader peptide. Right? It is not a true protein because it doesn't have enough amino acids to form tertiary structure and form a compact structure. It was just kind of a random coil piece of polypeptide that will float around within the cell. It is not considered a structural gene because it is involved in the regulation of attenuation. And one of the key things to point out here is that there are actually two codons for tryptophan within the trip leader peptide. And remember that tryptophan is the product of this operon, right? The whole point of this, the trip operon, is to biosynthesize tryptophan. Tryptophan is, even at its normal concentrations within a cell, is a relatively rare molecule. It's the rarest of the amino acids. So in this stretch of amino acids, the fact that there are two tryptophans is remarkable. And the other thing to notice here is that the mRNA sequence has the ability to form stem loop structures. And in fact, there's actually a couple of different alternate stem loop structures that can be formed. You have, and they're often highlighted as three different regions: region 1, region 2, region 3, and region 4. So this is 1, 2, 3, and 4. And the idea is that depending on which stem loop structure is allowed to form, you will have either attenuation, so a termination of transcription, or you will not have termination. So this is often referred to as anti-termination. If the 3-4 loop forms, that is actually a row-independent terminator loop. Right? It is immediately followed by an attenuator sequence, right? So that's a poly-U region, which means on the template DNA, the complementary sequences are poly-A. and that is where those are characteristics that are found in row-independent termination of transcription. This site over here represents the beginning of the first structural gene, which is TRPE. So you can have termination at this site after these U's are transcribed. When attenuation occurs, if attenuation does not occur, transcription proceeds. And it's very important for attenuation. This is a mechanism that is only found in prokaryotes because it involves the coupling of transcription and translation. So let's look at this in a bit more detail on the next slide. But before we do that, as a reminder, because we have these two trip codons, they are going to effectively function as a mechanism by which the cell can sense the concentration of tryptophan within the cell. So, as I mentioned, here's another representation of that messenger RNA that's transcribed from the 5' end all the way around to the 3' end. There's our attenuator sequence. This is that 3-4 loop structure that's potentially formed, and this is the 2-3 loop structure that's potentially formed. You should keep in note that even though it's represented here as both of the loops are forming, that's actually not possible. Only one of those two possible loop structures can actually form, right? Because this G, if it's having complementarity with this C, is not capable of also having complementarity with this C, right? The hydrogen bond donors and acceptors are already occupied. So basically there's a choice between forming this stem loop and this stem loop region. Right? So when the 2-3 loop forms, right? So this region and this region form, right? The bases within the 3 region are sequestered already by base pairing and are not available to interact with the 4 region. So as a result, you cannot form the 3-4 loop, and these are conditions under which you have anti-termination, so no termination occurs. If for some reason this 3 region is available when the 4 region is transcribed, that's when you form a stem loop structure which forms that terminator loop, and you get attenuation or termination of transcription occurring. Right? And so remember that this is being transcribed in sequence, right? This 2 region is formed before the 3 region, and the 3 region is formed before the 4 region. So unless there is something interfering with it, this 2 region will be available to interact with the 3 region, and therefore the 3 region should not be able to interact with the 4 region. So this was kind of the state that occurs unless there is some sort of intervention. And that intervention comes in the form of the ribosome. So while transcription is occurring, translation is also occurring at the same time before transcription even ends. You're already translating the leader peptide before you even start transcribing that first structural gene. And so it's this linkage of transcription and translation that allows attenuation to occur. So the idea is that the ribosome, when it's translating that leader peptide, is going to have a footprint on the RNA and can prevent some regions from interacting with other regions. So if the ribosome, for example, is allowed to translate the entire leader peptide, its footprint will include portions of that 2 region, and as a result, the 2 region will not be available to interact with the 3 region, and therefore the 3-4 termination loop can form. Alternatively, if the ribosome does not proceed efficiently, if it stalls within the trip leader peptide, the 2 region does become available to form a stem loop with the 3 region. The 4 region does not form a stem loop with the 3 region, and you have anti-termination. So transcriptional elongation proceeds. So let's look at a picture here. This kind of boat whale thing represents the ribosome. We have conditions where we have low levels of tryptophan. What happens is the ribosome will initiate translation at the start codon. It will translate 1 codon at a time, but when it reaches the first trip codon, what will happen is, because tryptophan levels are low, tRNAs that are connected to tryptophan, so tryptophanyl tRNAs, so those are tRNAs that are aminoacylated to tryptophan, will be at very low concentrations. And as a result, the ribosome will have to wait until a tryptophanyl tRNA diffuses into the correct site of the ribosome. And once that finally occurs, the ribosome will index to the next codon, which is yet another tryptophan. And when that and then it will have to wait again until a tryptophan diffuses into the correct site of the ribosome to allow it to continue to proceed. So as a result, the ribosome stalls at this tandem trip codon within the trip leader peptide, and the RNA polymerase will kind of continue to motor along its merry way and continue to transcribe the rest of the mRNA. So as the gap widens between the ribosome and the RNA polymerase, which would be over here, the 2 region is exposed. When the 3 region is then transcribed, it interacts with the 2 region, forming that 2-3 loop. And then when the 4 region is transcribed, because there is no 3 region available to form the stem loop, you have no termination occurring. So you're allowed to elongate. And that makes a lot of sense from a functional point of view. At levels of low tryptophan, you want to express the trip structural genes to make more tryptophan. If we look at another representation of this, again at low tryptophan levels, right, this shows the coupling of transcription by RNA polymerase and translation by the ribosome. So here's the template DNA, here's the nascent RNA transcript, and here's the ribosome. The ribosome hops onto the leader peptide start codon, stalls out here, allowing the 2-3 loop to form. The 4 region therefore has nothing to complement to, and as a result you get anti-termination or continuation of transcription. All right, so when you have a non-starved situation, so that means that there are abundant tryptophan amino acids present within the cell, we now have our kind of blobby whale, which is the ribosome, is proceeding beyond the tryptophan codons because it doesn't stall there, because it doesn't have to wait for the rare tryptophan amino acid to appear. Instead, it can diffuse in fairly readily. And as a result, the 2 region, the footprint of the ribosome when it reaches the stop codon includes part of the 2 region. As a result, the 2 region is occupied when the 3 region is transcribed, does not form a stem loop structure. Therefore, when the 4 region is transcribed, it does form this termination loop, this row independent termination loop, and you get transcriptional termination at this attenuator site. Right, so again, when we have abundant levels of tryptophan, we get termination of transcription, and you do not get expression of the trip genes, which are needed to make more tryptophan. And you don't really want to be making them anyway because you have enough tryptophan to begin with. Another representation, same scenario, tryptophan levels are abundant. We get less of a gap that forms between the RNA polymerase and the ribosome. The ribosome makes it all the way to the termination codon, sits on part of the 2 region, allowing the 3 and the 4 region to form that termination loop, and therefore we get transcriptional termination or attenuation. Right, so sufficient tRNA, etc. Right. I've included this slide once again to kind of put in text a lot of the information that I have provided via comments in the previous slides. Right, so we'll end with a conceptual question. So imagine that a mutation occurs where these 2 trip codons are converted to a different sequence, tyrosine, for example. What does this do to the levels of trip expression, assuming that tyrosine levels are high? So think about that before you proceed to the next slide. So If tyrosine levels are high, that means the ribosome is going to proceed beyond the previously tandem trip codons all the way to the stop codon. Its footprint is going to occupy part of the 2 region. That allows the 3,4 termination loop to form, and as a result you get termination of transcription, and that prevents expression of the trip structural genes, including TRPA, and therefore you see an overall decrease.